Hello and welcome to Nikolai's genetics lessons, and today we are going to talk about basics of forensic and population genetic statistics. Imagine that we have a pair of chromosomes, and this is homologous chromosomes. One we inherited from the mother side, another from the father side. And now imagine that here we have certain locus, and in this locus, in other words, locus is just position on the chromosome, in the same position on two homologous chromosomes. Of course, we are going to have the same gene. And it's not necessarily have to be a gene, it can be any sequence, DNA sequence that we are using for our own purposes. And for example, in forensic genetics, we choose such loci which just provide us different variants of the STR or short tandem repeats. This is such fragments of the DNA that highly variable. So sometimes we can find such allele with repetitive DNA, which say there is a sequence A, T, T, G, may repeat 5 times, sometimes 6 times, sometimes 20 times. So we may have many different alleles with different number of repetition of the same sequence. So I would put N here. And each allele just would represent a fragment of the different lengths. Because if it would be repeated 10 times, the length would be different if it's going to be repeated 20 times. So now imagine that in this certain locus we have different alleles. Let me show here in the cloud different alleles. Let's say allele A, allele B, allele C, and allele D. And each allele, of course, can be present in the human gene pool with different frequencies. For example, allele A can be present with the frequency of 0.1 or 10%. Allele B can be present with the frequency of 0.2 or 20%. And say allele C would be present with the frequency of 0.3 or 30%. And let's combine 10, 30, 60. So, and allele D would be present with a frequency of 0.4. All alleles in the gene pool combined, if we combine the frequencies, we should get 1 or 100%. So now we have 4 alleles which represent 100% of the alleles for this locus with different frequency of the occurrence. Now the question, what is the probability that in this locus we will find 2 alleles A, or for example allele A and B? It seems like very simple question, and many of you would say that calculations are going to be as follows. 0.1 frequency of the allele A, we have to multiply by a frequency of the other allele, which is also the same allele A, which is also 0.1. And the answer is going to be 0.01. Or if we need an answer in percentage form, we just have to multiply this number by 100 and we are going to get 1%. So probability that here in this locus we would have 2 alleles A would be 1%. Now as for this variant here, many of you also would say that calculations would be as follows. So frequency of the allele A is 0.1. We have to multiply by the frequency of the allele B, and this is going to be 0.2. And the answer should be 0.02. But this is actually wrong answer. So at this locus here, we can get actually two combinations. On the left chromosome, which is say paternal, we can get allele A, and on the right chromosome, which would say maternal, we can get B allele. But also another variant can be that on the paternal we can get B allele, and on the maternal we can get A allele. So two combinations are possible, and by the way, AB is equal to BA, because we don't have left and right inside the nucleus. So our calculations is going to be, we have one combination A, B, and we have another combination B, A. So we are going to get 2A, B. And for those people who are still confused, let me change letters to frequencies. For example, frequency of the allele A is 0.1. Here's multiplication sign. We have to multiply by the frequency of the allele B, which is 0.2 plus 0.2 multiplied by 0.1. So what we are going to get here, we are going to get 0.02 and 0.02 here. So our answer is going to be 0.04 or 4%. Again, if you need an answer in percentage form, we just have to multiply this number by 100. And this is going to be an answer in percentage form. So our calculations actually have to be 2 multiplied by 0.1 and by 0.2. And the answer is going to be 0.04. Also give you another explanation. Maybe it would be more clear for you. So we have 4 alleles in our gene pool. Allele A plus allele B plus allele C plus allele D. And this is our gene pool in this parenthesis. And here is in a power of 2 means that we have a diploid organism, and all combinations would be possible in diploid organism. So in a sets of 2. And how we can expand this formula? We can expand as follows. We can say that this is the same as allele A plus allele B plus allele C plus allele D multiplied by allele A plus allele B plus allele C plus allele D. And this would equal to A multiplied by A is going to be A squared. A multiplied by B is going to be AB, so plus AB. A multiplied by C is going to be AC, so plus AC. A multiplied by D is going to be AD, so plus AD. Next, B multiplied by A is going to be BA, so plus BA. B multiplied by B is going to be B squared. B multiplied by C is going to be BC. B multiplied by D is going to be BD. C multiplied by A is going to be CA. C multiplied by D by B is going to be CB. C multiplied by C is going to be C squared. C multiplied by D is going to be CD. And lastly, D multiplied by A is going to be DA. D multiplied by B is going to be DB. And D multiplied by C is going to be DC. and d multiplied by d is going to be d squared. Now let's simplify. So we have here a squared, we have here b squared, we have here c squared, and we have d squared. So a squared plus b squared plus c squared plus d squared plus, now take a look, we have ab, but we also have ba. We can put it as 2ab. We also have ac, but we also have ca. So we can put it as 2ac. Next we have ad, but we also have a da. So we can put it as 2ad. We also have here bc and cb plus 2bc. Next we also have bd and we have db. So plus 2bd, and the last would be cd and dc plus 2cd. So this is our final formula, and you see that whenever we see homozygotes, we just see that it's squared. Homozygous for two alleles A would be just A squared, the same for B, C, and D, but all heterozygotes, which can be AB, AC, AD, BC, BD, and CD, have this sign here, so we have to multiply by 2. That's why here we also multiply by 2. So I hope now it would be much more clear why we have to multiply by 2 when we multiply probabilities of heterozygotes. So when we see homozygotes, we just do simple multiplication. When we see two different alleles, we also have to multiply by 2. And this is very, very important for your understanding of this basic principle concept idea, how statistics works. It is very important for genetic profiling, for forensic cases, for example, for cases like paternity testing, and many, many other population genetics applications. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.